Hello students, so in last video we have discussed about the definition and history of pharmacognosy. So in the same we are continuing it here, the scope of pharmacognosy. So when it will be asked in exam what is the scope of pharmacognosy, what are the points you are supposed to describe, and you should mention in a proper example, right? So in scope of pharmacognosy, generally what is the meaning of this actually when we talk about scope means what are the in which are the area where its utility is there, like for example scope of B pharmacy. So whenever you might be knowing that so many of your family friends and neighbor, they will be asking you, oh, you are doing B pharmacy, so what are the scope? What you are telling after B Farm, what you are going to do, hospital pharmacist, drug inspector, medical store, those are the scope you are telling where you can be placed. So in the same way we can understand here also the scope of pharmacognosy means the what is the utility of these subjects into the pharmaceutical science, where we can what are the use we can gain from these particular subjects or this particular knowledge. Okay, so the first thing it is there, the first isolation or analysis of phytochemicals or phytoconstituents, right? As we know that the phytochemicals in history part we have seen that nicotine from tobacco, morphine from opium, right? So like that here also digitoxin from digitalis leaf, or maybe glycoside from digitalis leaf, or atropine from belladonna, or alkaloid from belladonna, right? So many chemical constituents of phytochemical it will be there into the natural drug or maybe plants materials, right? From where we can isolate, because in history we have seen that lots of contribution has been done by the different scientists into the different time period, and they have successfully isolated the phytoconstituents from the different chemical different plant materials, right? So these things can be done into this particular subject area. The second thing, second scope you can write down is structure activity relationship, right? So it is nothing but it is also known as SAR, right? You are going to have in medicinal chemistry also SAR, right? So when we talk about structure activity relationship, as the name indicates, structure is important for the activity, and these two relationship are very important to give therapeutic value. Okay, the theme is like that only. So example what you can write here, hypotensive activity, HYPO, hypotensive action of reserpine are due to the trimethoxybenzoic acid. Means to have hypotensive action of this particular chemical constant reserpine which is obtained from Rauwolfia, this is the trimethoxybenzoic acid is important, right? So in each and every structure the each and every bonds are playing major role, right? If you remove this bond, either the activity may increase or it may decrease. It depends on the what kind of structure. That's what here in example generally Rauwolfia anti-hypertensive, right? So because of this trimethoxybenzoic acid is very important, this in the structure this particular thing should be there, then only this reserpine will be giving the hypotensive activity. That is the meaning of this. Third point what you can mention here is drugs obtained by partial synthesis of natural products. Okay, so what is happening from the natural products? When we do partial synthesis, the drug can be obtained. For example, steroids, hormones can be obtained from diosgenin, right, by acetylysis. So this example you can mention in your what you call answer. Now coming to the fourth one, what is that? Natural products as model for synthesis of new drugs. Okay, so what is the meaning of this heading? The natural products or natural drugs are the important, or they are going to be the model. From there only, for by seeing them, what is the structure, what is the kind of things, bond, side chain, we can synthesize the new drug similarly having the similar action. For example, morphine is the model for the so many potent drug. Like morphine, what are the things which will be there, like hydroxyl group, tertiary nitrogen group, and what do you call, that's what in morphine structure we are seeing. No, ether bridge is very important. Ether bridge is very important. So whenever the potent drug we are going to synthesize in our laboratory, this particular if you are expecting the analgesic activity as morphine, this ether bridge has to be there, tertiary nitrogen has to be there. So like that it become model. Means if you are expecting the analgesic activity, morphine like structure supposed to be there in our synthetic drug. Okay, that is the thing. Fifth one, drugs of direct therapeutic use. like antibiotic we have to use directly, vincristine, right? So there are so many examples. For that there is no replacement, we have to depend on to the natural sources only. Biosynthetic pathway investigation, that's also, see, chemic acid pathways there, acetate hypothesis can be done by this particular thing, right? So these are the 6 points under scope, and further we can mention further few points which is important into the scope. That is, after 6 point, the 7 point we can mention here is cultivation, right? Cultivation and collection of medicinal plants. Okay, so as we know that medicinal plants if we are going to cultivate, we are going to see their altitude, rainfall, temperature, irrigation, soil fertility, and so many factors we are taking care, and we are focusing on to the maximum yield, maximum percentage yield, for which chemical constants we are cultivating that medicinal plant. For example, in clove, eugenol; in cinchona, quinine; in cinnamon, cinnamaldehyde; in opium, morphine. So these cultivation can be ensured the with the maximum percentage by the having these particular things, right? 8 point we can write preparation of herbal formulations, that is only possible into this particular field, like preparation of churnas, different types of ashwas, arishtas, leha, that can be done. And the 9 point you can mention development of plant tissue culture, right? Like citrus species can be developed, solanaceae plant can be what you call have proper by having the proper micro propagation we can get the proper plant, and with the good percentage yield. Now after scope, we can mention here is the development of pharmacognosy, very very important, right? That is the last point under in the first we started in first video, that was the uh, definition, history, scope, and development of pharmacognosy, right? So definition and history we have discussed in the last video. Just now we discussed the scope, now development, right? So when we talk about the development of pharmacognosy, we can divide it in the four category or four part. Those four part are the before the 19th century what happened, then early modern time what happened, then modern time what happened, and the 20th century what is happening. Okay, so now development, these things in this point you can write down. The first one, before the 19th century, what kind of development occur into the field of pharmacognosy, that we are going to write. For example, medicines originated in Egypt and India, right? In Egypt, we have discussed the papyrus ebers, right, in the history also, 1500 before Christ. So and in India, Ayurveda we have discussed, like 3500 BC. There was a Riga Veda and Atharva Veda, right? From there Ayurveda developed. So in Egypt, papyrus ebers, there was a description about crocus, dried ox bile juice, and castor oil. These things it was there, right? So that time itself it started. Then in 77 after death, Dioscorides revealed about 600 crude drugs in his De Materia Medica, that we have seen. Charak Samhita, Shushruta Samhita, we have seen Charaka. Charak has done the 50 group of the 10 herb sets. Then Shushruta, he has did 760 herbs in 7 distinct sets, right? And the property, what are the property, according to that he has divided, right? Then Pliny in 23 to 79 AD, 1000 species of the plant was compiled. So these things you can write down before 19th century, right? Now when we go further, second point which we can mention, that is the early modern time, right? Or in early modern time, what we can write here, 1815 to 1930. So what kind of development happened or occur at that period of time? C.A. Sadler, we have seen in 1815, he written one thesis, doctoral thesis, Analecta Pharmacognostica, and from there only the term the term pharmacognosy it was revealed. Then Derosen in 1803, he isolated nicotine from opium. Then Serturner in 1806, morphine from opium. Then qualitative and quantitative methods has been developed in those period of time. Crude drugs identification we started, right, in 1815 to 1930. So many identification, like Nux vomica seed, like what do you call opium, latex, so many things we started identifying. Fluorescence analysis also came into the picture. Chromatography also, thin layer chromatography also started, like how we can separate them. Column chromatography is started during that time. Pharmacognosy became one of the independent subject by the end of the 1930, right? Then the further if you go, that is the next one, that is the modern time, right? So when we talk about the modern time, we are we are having 1930 to 1990, right? So this period we have mentioned here. 1930, the development of biology and chemistry taken place, right? So in these two subject, lots of development occur, and the bioassay of crude drugs also started, right? So what is the action of drug action, and they started doing into the different animal models, rat, mice, guinea pig, monkey, and all those things. Animal model started using in this period of time. Study of active ingredients, what is the kind of structure it is there, what is the absorption, distribution, where it is going to metabolize, etc. Then we had so many of instrumentation techniques like colorimetry, spectrophotometry, isolation techniques. For example, Reserpine from Raulfia and Vinca alkalite has been isolated in this period of time. Like then fourth number what you can write down, it is here is early 21st century now, right? Early 21st century, in this we can write down the separation of constituents taken place, structure determination also started. Now we are having qualitative technologies for the ensure of the particular drug or medicament. We are having 1H NMR, 13 carbon NMR, DNA fingerprinting, right? So lots of advanced technology has been developed to know about the structural and genetic level of the particular constituents. In modern concept, we are bothering about the isolation and the structure activity relationship, because we are having lots of chemical constituents has been identified. Now we are trying to increase their potency by using the different technologies, right? So this you can write down in the development part with the effective manner, proper example. Thank you.